Hello, everybody. Welcome to a new episode of the Dissenter. I'm your host, as always, Ricardo Lopes, and today I'm joined by Dr. Jessica Nowicki. She's a research scientist at the Wu Tsai Neurosciences Institute at Stanford University. She is interested in the mechanistic basis of prosocial behavior in early vertebrates, and today we're going to talk about that and about pair bonding, empathy, and other related topics. So, Dr. Nowicki, welcome to the show. It's a big pleasure to have you on.
Thank you. Thank you for having me, Ricardo.
So, prosocial behavior then. How do you study that from an evolutionary psychology, and what is it basically?
Well, that's an excellent question. It's basically any behavior that is voluntary and is aimed to, intended to benefit another individual. So either by relieving stress in that individual or improving their well-being. And so we're talking about positively valenced behaviors, helping, reciprocity, cooperation. These are more sophisticated examples, but there's also very simple forms of prosocial behavior, like just side-by-side contact, huddling with each other, allo grooming. So, just combing through the fur of a companion in order to ease their stress, or if you're a bird, it would be called allo preening. And from an evolutionary perspective, this has evolved not out of no reason. There are good reasons why it has evolved. There are adaptive benefits to it. So not only does prosocial behavior affect and help the individual who's receiving it, but also the giver ultimately. And so this behavior is not fluff or trivial. It is selected for, and it can be very adaptive.
And do we know how early in evolutionary history did it arise? Do we know how old it is?
Well, I think it's important to consider the background of the study of prosocial behavior, the historical background of it, and that is very human-centric. And so for a long time, the paradigm, the way of thinking was that prosocial behavior was, unique to humans. Think the scala naturae, I think it's called, this hierarchy of beingness across humans and other animals. And on top of the hierarchy, you have humans, they're closest to God, and therefore we have been endowed with these special attributes, and one of them is morality and prosociality, and that's what makes us so special and unique. And so for a long time, that way of thinking, which we now know is ridiculous and flawed, integrated the sciences as well. And people knew better to look for prosociality in anything but a human. In fact, the word prosociality and humane are almost used synonymously because we believe that the two were so inextricably linked to each other. And in contrast to that, non-human animals were inhumane or beastly. And so for a long time, nobody looked for prosocial behavior, and we thought it was a unique trait. But that's just simply not true. One of my favorite examples of that is in my own life experiences. I was doing my PhD, and I am a coral reef biologist. So I was out in the field on an island with a legend in coral reef biology. He had been doing that. He was into his 70s. I mean, the field wouldn't be what it was without decades and decades of his work. Yeah. And he and I and another young PhD student were sitting there, and this PhD student was describing, he said, I think I'm seeing this weird behavior between these two coral reef fish, these rabbit fishes. One, it has its head pointed down into the reef matrix and is foraging, and there's this other one right next to it, and its head is sticking up into the water column, kind of looking around, like as if he was looking for predators or something. And And then when the forager is done, they take turns and they switch back and forth. And this legend said, no, I would have, that doesn't exist. I would have seen something like that had it been there. And the next day he goes out into the reef when he's doing his regular, research that he had planned to do. And that night, the next night, we were all conversing over the barbecue, and he said, God, you were right. He said, I saw it. It was right under my nose this whole time. And this has now been published several times and described very well. This behavior, this prosocial behavior, is called reciprocal vigilance behavior in rabbit fishes. And not only was it under his nose, but it's everywhere. Like, it's all over the reef. These fish are doing this. And so I think that's just a case example to show that not only is it everywhere in fishes, but it's everywhere all over the animal kingdom. You can find different forms and varieties of cooperation in all sorts of social systems, not just pair bonding, even in solitude organisms that live their life most of the time by themselves. And in every major lineage of vertebrates, you can find prosocial behavior. It's even in insects. So there's monogamous and pair bonding beetles that have these, highly cooperative biparental care strategies. There are huntsman spiders that are amazing mothers that care for their little spiderlings on her back. And crustaceans, there's, little shrimp that go into clients' mouths and will clean the teeth and pick the parasites out of the client's mouths, which obviously benefits the client, but it also benefits them too. They get food out of it. But the client, if it's pleased, it'll keep on coming back for more services. And so this is definitely a beautiful reciprocal prosocial behavior between the two. And I was watching a really cool documentary the other day called Fantastic Fungi, and they were describing a fungus called mycelium that lays these networks, these highly integrated networks, into the soil through which trees can passage communication signals through each other in order to communicate, in order to recognize their kin, in order to share nutrients. I mean, this is behavior that is helping another individual, whether it's voluntary and whether it is intended to do that, I guess it's arguable. So that's, still debatable, but it certainly is helpful. And what we know for sure now is, far from being unique to humans, prosocial behavior is ubiquitous across at least animals. And it's hundreds of millions of years old, and it's conserved.
Yeah, so at the very least, it's safe to say that it's very old in evolutionary history.
Yes.
Yeah. So, how about affiliative behaviors? What is that?
I think affiliative behaviors are just prosocial behaviors that regularly occur between the same individuals. So, as opposed to a prosocial behavior that is expressed to a stranger just once and then never happens again to that same individual. And that's kind of why they call it affiliation. And I think most prosocial behaviors are considered affiliative. So, for example, I'm trying to think of a good example here. Cleaner fish there's these little wrasses on the reef. They will set up these cleaning stations, and the resident fishes on the reef, they know where these stations are, they know who these cleaner fish individuals are, and they return time and time and time again to the same place. And they develop relationships with each other that require trust, because sometimes the cleaner fish, if it goes into this resident's mouth, it might be eaten. And likewise, the cleaner fish sometimes can take a little nip out of the yummy mucus of the client, and they could hurt the client. And so there has to be some level of trust happening there. And that is a prosocial behavior, and it's ongoing through time. And so I think that's one example of many, many affiliative forms of prosociality.
And still about affiliative behaviors, of course, it's also important to understand some of the mechanisms underlying these kinds of behaviors. And do we know if across species there is convergence when it comes to the mechanisms that underlie affiliative behavior or not?
I think overall, yes. I think it's important to first consider that it's possible for convergence to even happen on a mechanistic level, on a brain level. Because, for example, if brain region X that we know mediates prosociality in species X doesn't even exist in brain region, in species B, then it's impossible for convergence to happen. But of course, we know now that that is not the case. We know that the neural components of social behavior in general are ancient. They were already established before in the common ancestor of vertebrates. And so these brain regions and the neurochemicals that signal within brain regions we also know are evolutionary ancient. Some of them, in the case of oxytocin, for example, forms of it exist in annelids and little worms. So it's possible. And if the affiliative behavior that you're considering is conserved, so if it was there in a common ancestor and it never went away as vertebrates, for example, evolved and diversified, then it would be really easy to conceive that, well, the wheel isn't broken, don't fix it. Like the mechanism is already there, just maintain it. Why suddenly switch to something different? So the idea of conservation of those underlying mechanisms is very possible. But when you're talking about converged behaviors or prosocial behaviors, for example, in the case of pair bonding, which I study, or parental care, this is a little bit different, these types of affiliative behaviors, because they have independently arisen hundreds of times, and sometimes across very vast evolutionary distances. So in other words, across species that are very distantly related to each other. And each time that happens, there's the opportunity for something different to happen. So I think you have to consider what type of affiliative behavior you're talking about and the evolutionary history of that affiliative behavior. In the case of pair bonding, even though it is a highly convergent trait across very diverse organisms, we nevertheless find that there appear to be core mechanisms involved, including one brain region is the amygdala. So the amygdala is important for emotional processing. It's important for conspecific recognition. And you can imagine these are all really important for bonding to another individual. You need to know who you're bonding with. And there has to be some sort of presumably emotional valence, a positive emotional valence associated with that bond. And so it makes sense that the amygdala appears to be repeatedly involved in pair bonding. And we also find some ancient type of neurochemicals, the nonapeptides, oxytocin, vasotocin, their ancestral homologs in fishes, and dopamine are repeatedly involved in the independent evolution of pair bonding.
So you're talking about pair bonding there. What is pair bonding exactly from an evolutionary perspective?
I Pair bonding is a type of affiliative or prosocial relationship, but it's specific in a few ways. So it is selective towards just one other individual, and it's enduring through time, and such that you can't just have for example, a male that is affiliating with a female just temporarily while she's in heat and mates with her and then leaves right after that. That's not a pair bond. It has to be some sort of enduring relationship that is overall prosocial in its nature and is selective between those two individuals. So a harem, for example, where a male or sometimes, in some cases, females, but mostly males have several females. They have several, I guess you could argue, bonds, but that's not a pair bonded relationship because there's several of them. And for a long time, researchers kind of interpreted the existence of pair bonding through a very traditional westernized perspective. And projected that idea onto nature, and that's what we would look for when we were, defining a pair bond. So what does that mean? That means, a bond between a male and a female, and a bond that involves sexual monogamy, and one that often involves biparental care. So both mom and dad are caring for the offspring. And this is the case in some instances in nature. You will find species that fall into this definition. Canadian geese, poison dart frogs, they are some examples of this. But we also find that in many cases, pair bonding has little, if not nothing, to do primarily with reproduction. And so ring-tailed lemurs, for example, they have very strong pair bonds, but they're sexually very promiscuous. And in other cases, an organism that I studied, I spent a lot of time focusing on during my PhD, is coral reef butterfly fishes. These guys are extreme oddities because they have very strong pair bonds that can last for decades. One pair was followed, I shouldn't say decades, but one pair was followed for around a decade. And, sometimes they only live for a decade, if not a couple. So the bond is very strong, but they don't exhibit any parental care at all. They just spawn into the water column. And sometimes you'll see male and male bonds and female bonds. And so there's something else going on here. And sometimes the bonds will be between sexually immature individuals altogether. Something else is happening. And so in the case of coral reef butterfly fishes, what we find is that they bond because of the need for joint territory defense. So in their case, butterfly fishes, they rely on a diet of coral that's very nutrient poor. And so in order to get enough nutrients, they're constantly feeding all the time, and they need to spend a lot of time feeding. But when you're feeding, you can't be defending your territory all the time. There's only so much time in the day. And so the best solution to this is to do it as in a team, in a partnership. And so individuals will team up. Usually it's male and female, but sometimes it's male, female. And they'll establish a territory. And depending upon the species, sometimes the female will spend the most of the time feeding while the male does his rounds around the perimeter, defending the territory and feeding along the way. And in other species, the pair bonds will do their rounds around the territory defense, in one cohesive unit. So this beautiful coordinated swimming. And it's almost like an advertisement to neighbors. They know who their neighbors are, and they're advertising to each other. Look, we're here, there's two of us, maintain your distances. And that's most of the time respected very nicely between different pairs. And so, that is a case where pair bonding is not fall into the traditional sense. Shingleback skinks are the only known species of reptiles to be pair bonded. And here they appear to be monogamous because they're subjected to a lot of parasites. And so there's a lot of parasite load in them. And what they find is if they're sexually devoted to just one other individual, they're less likely to be infected as opposed to individuals that are more sexually promiscuous. So lots of different selection pressures for pair bondedness, lots of different forms of it, and ways in which it can serve an adaptive function.
Mm-hmm. And you've also studied pair bonding in poison frogs, correct?
Yes. So in poison frogs, there is a species called Ranitomeya imitator. And here, so they exist in Peru. And I personally haven't done this work. This is decades of work from other groups that have done an amazing job classifying the way that they live. And so males and females will join up and they will create a bond together in a shared territory. And in this territory, the females will spend most of their time foraging, again, because they have a very high energetic demand. Eggs are expensive to make. And so they'll spend a lot of their time foraging, and the males will guard the developing embryos that they have and kind of wait for them to develop enough to the extent that they're ready to hatch. And when the embryo hatches, the male will kind of assist it by using its hind legs to kind of break open the little and help it get onto its back. And sometimes they'll just wiggle their own ways on top of the male's back. And then the male, piggyback style, will give them a piggyback ride over to a little pools. And they'll spend, and this is within the territory, and they'll spend a long time and travel long distances within this territory in order to find ideal pools for the little developing tadpole to finish its development. And so he deposits it into this pool, and then he proceeds to babysit, more or less, this little tadpole while mom is out there foraging. And at some point, the tadpoles get hungry, and so they will, what we could call, beg to dad. And so this involves a behavior where they just basically gyrate their head against the little froggy feet of dad that are dangling there in the water. And he will then call out to mom, and mom will stop what she's doing, and then she'll come over to the tadpole and she'll deposit one, sometimes two unfertilized eggs, we call them trophic eggs, into the water column so that the little tadpole can feed on these. And they're highly nutritious. And they will do this for several tadpoles, developing embryos simultaneously at the same time, and over the course of months within a reproductive season. So this is very sophisticated, hard work, teamwork between the parents. This is biparental care, and it wouldn't be possible without each other. So this is one of the reasons why pair bonding has evolved in the species of poison frog.
By the way, earlier you mentioned at a certain point that, for example, the amygdala in the brain is associated with pair bonding, for example. Do we know what are the genomic and other neural bases of pair bonding and parental care?
In terms of the overlap of them two, like what genomic or neural components overlap between those two bonds.
Yes.
So most of the work on that has been done in female rodents. Um, and the idea there was that there would be a strong overlap between those two mechanisms because in female mammals, we all lactate. And so that our offspring rely on milk to survive. And so there's always a maternal bond there in female mammals. And so the idea is, well, every time pair bonding then independently evolved, which because it independently evolved, it's not always there. But every time it did, well, wouldn't it make sense for evolution to recruit that maternal bond that's already there in order to mediate the pair bond? I mean, keep it simple, right? And in fact, that is what we find in several ways. There's again, the amygdala and the nucleus accumbens are repeatedly involved in both types of bonds, at least in female mammals. And so too are nonapeptides and dopamine.
Mm-hmm. So talking about one last type of prosocial behavior or emotion in this case, I know how it is classified exactly, but empathy. So what do we know about it from an evolutionary perspective?
Well, I think it's important to first define empathy and then also consider, why it even exists, why it might exist in the first place.
Yeah.
And so empathy can be defined as the ability to embody or take on the emotional state of another individual. And when considering prosocial behaviors, I had previously said they have evolved not only because they confer some sort of a fitness benefit to the receiver, but also the giver. And so if the giver of the prosocial behavior is going to put in the effort to do that, there has to be some reason why, some motivation. And surely it can't be because they understand the adaptive significance of that. Going back to the example of the poison frog mother, she wouldn't like sit there for days doing the calculation, being like, okay, so if I give you one trophic egg, I might have 2 offspring that survive, like carry the one. Okay, this is going to be a net benefit sort of a thing. Like that does not happen.
Yeah.
And so there has to be a much more simple motivation. It has to be simple. It has to be immediate. And that is the power and function of empathy. And so, in the case of the mother and her begging tadpole, she sees her tadpole in distress. That, in theory, that elicit distress in herself through her empathetic connection. And she wants to alleviate that distress in the tadpole in order to alleviate it, and as if she was alleviating it in herself. And so she helps the tadpole. And so that is kind of the conceptual idea behind empathy and how it drives cooperation, reciprocity, helping, and prosocial behavior in general. But like prosocial behavior, no one was looking for empathy in non-humans. We thought it was strictly a human trait, one of those things that makes us moral and unique. Mm-hmm. And it wasn't until a couple decades ago when a famous primatologist, Franz de Waal, started to look for it in primates, and he found it in primates. And the whole world was like, oh my God, And then somebody found it in birds. And so we know that birds have a version of empathy called emotional contagion. And then it was discovered in fishes. And so I set out to determine whether an ancestral form of empathy called emotional contagion exists in amphibians, because no one had looked for it or documented it in amphibians, and I was studying a great model system for that. with, these pair bonded frogs, very strong bonds, the high level of cooperation. And so it's a good place to start looking. And we started a colony in our lab. And in order to study, the presence of empathy, I didn't know where to begin in the life of a frog. And so I used a framework. I borrowed a framework that was already established for rodents, that I just called the empathy essay. And basically how it works in rodents is in these pair bonded rodents, for example, the female will be subjected to a stressor, and then she will be reunited with her male partner. And then the researchers look to see whether the male partner himself shows signs of distress through observing it in the female, even though he never experienced or even observed that stressor himself. And sure enough, they find it in prairie voles. Males will match the cortisol state, the heightened cortisol state, the physiological signature of stress that their females have, and they'll also match stress-like behavior. And beyond that, the males will also show consolation behavior. So they'll do behaviors to try to calm her down, help her feel better. And so this is kind of the design that I borrowed for the frogs, not knowing at all what I would find. But a few things were different. The stressor that I used was a gentle leg restraint. So I just grabbed gently the hind leg of the female, and it's unpleasant. She doesn't like it. And then put her back into the observation terraria with her male partner. And I then measured the cortisol and behavior of the male and the female. And what I found was that, surprisingly, the stressor did not stress the female. So her cortisol levels were not increased. That I was disappointed by from an experimental, obviously, in one way it's good, but from an experimental perspective, it was not ideal. But what I was equally surprised to have found was that the males still exhibited emotional contagion towards their female partner. And so they still matched her cortisol state, stress level, even though she wasn't stressed by it. So if the female partner happened to have been stressed for whatever reason, he increased his stress level, and vice versa. If she was low, then he would be low. So there was this physiological state matching happening with stress. So that was very exciting. And what was equally interesting was that this emotional contagion response was specific to the female partner. So when I repeated the experiment with females that were very similar to the female partner, same size, same reproductive state, the male didn't state match the non-partner female. And so this showed that his empathetic-like response is tailored, is specialized for her. She's important to him in some way. And beyond that, this response was irrespective of the amount of time that they had been bonded together. So some of these bonds I'd only had established for a few months, others for years. And so that emotional contagion was always there, and it was there irrespective of how many offspring they had had. So their reproductive history of their reproductive investment. So obviously this is a very strong response that I think warrants, opens up all these questions about why it's there and warrants deeper investigation.
Mm-hmm. So at the very least, we can safely say that empathy is much more widespread across the animal kingdom than just humans, just primates, or even just mammals, right?
And empathy in one form or another. There's arguably more sophisticated forms of empathy that involve state of mind, like understanding another person's state of mind from an intellectual perspective. That is thought to be reserved to, dolphins or humans. But at its very core, the most basic essence of empathy, which is emotional contagion, yes, that is, we're finding that's widespread phylogenetically, just as Darwin himself had speculated over, back in the 1800s.
Yeah.
And he was a bit of a renegade for thinking that. People thought he was a little bit loco, but no, he's right.
Yeah, back then he also had to be a little bit worried about religious backlash and all of that.
Absolutely. You got to be careful what you say.
Yeah, some of the claims he was making anyway were already very controversial. So.
Yes, and I should say that they still are. I had first became interested in studying trust and empathy in non-human animals back in 2012 when I was a young PhD student, and the idea was almost laughable within my scientific community.
So just 11 years ago it was still laughable?
Within my scientific community, yes, where I was doing my PhD, when I spoke to people who had been in the field for a long time, they said you have to be very careful because you could be exiled from science before you even get into it. There is a deeply rooted, I guess, tabooness that still resonates. I think it still lingers, but those constricted ways of thinking are slowly being broken down through persistent and I think courageous researchers.
Mm-hmm. Yeah, and we'll come back to this point in my last question because that's very interesting. But let me just ask you another question before that. So earlier when it comes to, for example, pair bonding and parental care, I asked you about the genomic and neural basis of them. But there's also gene expression, right? It's not just the genes or the genetic material itself, but also how the genes get expressed. So how important is it for us to study and understand how gene expression works in prosocial behavior?
That's an excellent question, and it's very important. And so unlike some morphological traits or pathological traits like, sickle cell is a classic example, or Huntington's disease, these traits can arise from single point mutations in the coding region of DNA, the part of the DNA that produces protein. And it's very pinpoint. It can be very pinpointed and simple as that. But in the case of behaviors in general, including prosocial behaviors, it's much more nuanced. And it's more the case where it's a interaction between several genes and specifically the expression of these genes that from which behavior emerges. And so, for example, in prairie voles, what they've found is that it is not the nucleotide sequences of, for example, a vasotocin, the vasopressin receptor that account for differences in bonding behavior, but rather, if you look upstream in the genome into the junk part of the DNA, which we now know is definitely not junk, you will find a little portion of the genome called a microsatellite, and it's just a small little chunk, and it helps govern the expression of the vasopressin gene itself. And so if there's variation in in the sequences of the microsatellite of a associated with the vasopressin receptor, then that changes the expression of the vasotocin receptor, which thereby creates variation in how that receptor is distributed and expressed across the brain. And the consequence of that is variation in pair-bonded behavior. So expression is very important. Another example was a study that looked across vertebrates, and it found that differences in gene expression on a whole brain level were attributed to variation in monogamy across vertebrates. And this was revealing because, like you said, it talks to the importance of gene expression, but it also created an opportunity for lots of follow-up studies, like, well, where in the brain specifically are these differences in gene expression happening to govern affiliative behavior, or in my case, pair bonding behavior. And so right now, what I've been doing for the last 6 years with an amazing team is to ask just that question. And so we are comparing the brains of pair bonded to solitary species and individuals within and across all of the 5 lineages of vertebrates, so fishes, amphibians, reptiles, birds, and mammals. And we're comparing the brains, specifically all the gene expression within the brain, and specifically within on a brain region specific level. And so this will help get to more fine-tuned understanding of the neural circuitries and the way that they are expressed in order to drive pair bonding behavior and variation in pair bonding behavior, and reveal, major mechanistic themes, but also deviations, how certain lineages might be unique from others.
So with that in mind, do you agree, for example, when certain people suggest that we should have an extended evolutionary synthesis, like people like David Sloan Wilson, Massimo Pigliucci, and others suggest, that we need to go beyond the so-called modern evolutionary synthesis to include things like gene expression or epigenetics and other stuff, like, for example, in the cases of humans and other species as well, culture, symbolism, and other kinds of mechanisms, let's say.
Absolutely. Yeah, absolutely. And this is a whole field that is up and coming, and it's booming, and I think it's just the tip of the iceberg. There are labs that are just explicitly focused on understanding epigenetic mechanisms of pair bonding behavior. That's all they focus on now, because it's so fruitful of a research avenue. Yes, the mechanisms are much deeper and complex than just simple nucleotide differences in the protein coding regions of genes.
Mm-hmm. So let me just ask you one last question then, and I'm asking you this because across the interview you've brought this issue up several times. So don't you think that we should be more wary, perhaps, as humans, of our own biases, and perhaps even more so our own cultural biases? Because when it comes to studying prosocial behavior and things like pair bonding, for example, just to stick with the example of pair bonding, we tend to have, particularly in the West, this very strict and limited understanding of, for example, what we would consider to be the nuclear family, with men and women, their children, and so on. And it's not even extended, because in other societies we have, of course, we know that we have cooperative breeding, and it's basically almost the entire family and community participating in rearing children and all of that. And it's not monogamy, there's also non-monogamy, and there's serial monogamy. It's not the idea that we just should stick with one person for the rest of our lives or something like that. So, I mean, that applies also to us humans, because that also gives us, unless you are an anthropologist, gives us a very limited understanding of how different or how varied human behavior can be across different societies, but also when it comes to understanding how certain behaviors and certain emotions can also be present in other animal species, for example. And of course, I'm not saying that there aren't any specific traits of humans that makes us different from other species. Of course, there are, and I don't even like to use too much the expression uniquely human, because that also implies some sort of essentialism, as if in the future, if we evolve into other species, suddenly then it would no longer be, I guess, uniquely human. And I'm even not sure, apart from language and perhaps very few things, what is really uniquely human. But I mean, do you agree with what I'm saying when it comes to really trying to go beyond our cultural biases when studying animal behavior in general?
Absolutely. Absolutely. I think that's what we're learning more than ever is, our conception of animal behavior. Historically, it's important to understand the history and how history has shaped our limited outlook. And it's important to, I think, recruit diverse types of people into the field of studying animal behavior, because especially groups of people who are indigenous and have a history of, very close observation of wild populations in their natural environment for hundreds of years. There's a lot of wisdom in that that Westernized science can and must learn from if we are going to advance ourselves. And so the more diversity we can recruit into science, and even in some ways naivety, fresh minds that haven't been conditioned to look for or be blinded to certain types of ideas, is critical if we're going to, especially if we're going to have major leaps in our understanding. And we're slowly getting there, and I'm very proud that the lab that I work with is, a part of that community.
Mm-hmm. So just before we go, would you like to tell people where they can find you and your work on the internet?
Yes, I've been very guilty of not finishing a web page for like 5 years. But you can find my contact information in a very brief bio of myself on the laboratory web page that I'm a part of. The laboratory for Stanford Laboratory for Organismal Biology. The PI there is Dr. Lauren O'Connell, and you can find me on that page, or the Wu Tsai Institute at Stanford. I'm alumni there, and so I believe I still have an active bio on that page. Yeah.
Great. So thank you so much again for your time, Dr. Nowicki, and it's really been fun to talk to you.
Thank you so much, Ricardo. Thank you for having me.
Hi guys, thank you for watching this interview until the end. If you liked it, please do not forget to like it, share, comment, and subscribe. And if you like more generally what I'm doing, please consider supporting the show on Patreon or PayPal. You have all of the links in the description of this interview. This show is brought to you by Enlights Learning and Development Done Differently. Check their website at enlights.com. I would also like to give a huge thank you to my main patrons and PayPal supporters, Per Larsen, Jerry Muller, Hans Fredrik Sunda, Bernardo Seixas, Olaf Alec, Jonathan Visser, Adam Kessel, Matthew Whittingbird, Arno Wolf, Tim Hollis, Henrik Alenia, John Connors, Philip Forrest Connolly, Robert Windager, Rui Inácio, Zup, Marco Neves, Colin Holbrook, Simon Columbus, Phil Kavanagh, Michael Stormir, Samuel Andreev, Francis Ford, Tiago Dunes, Alexander Dunbauer, Fergal Cusson, Hal Herzog, Nuno Machado, Jonathan Librant, João Linhares, Stanton T, Samuel Correa, Eric Hines, Mark Smith, João Meira, Tom Hummel, Sardas France, David Sloan Wilson, Yacila Dez Araújo, Romain Roach, Diego Londonho Correa, Yannick Punter, Adana Rusmani, Charlotte Bliss, Nicole Barbaro, Adam Hunt, Pavel Ostazewski, Nelek Bach, Guy Madison, Gary G. Hallman, Sam Afzal, Adrian Yegi, Nick Golden, Paulo Tolentino, João Barbosa, Julian Price, Edward Hall, Edin Bronner, Douglas Fry, Franca Bortolotti, Gabriel Panos Cortez, Ursula Litska, Scott, Zachary Fish, Tim Duffy, Sonny Smith, John Wiseman, Morten Eklund, Daniel Friedman, William Buckner, Paul George Arnaud, Luke Loaki, Georgios Theophanos, Chris Williamson, Peter Olsen, David Williams, Diogo Costa, Anton Erickson, Charles Murray, Alex Shaw, Amaury Martinez, Coralie Chevalier, Bangalore Atheists, Larry Dealey Jr., Holt Ericbud, Sterry, Michael Bailey, Dan Sperber, Robert Gressis, Tom Roth, DRPMD, Igor N., Jeff McMahon, Jake Zool, Barnabas Radix, Mark Campbell, Richard Bowen, Thomas Dobner, Luke Nissen, Chris Story, Manuel Oliveira, Kimberly Johnson, and Benjamin Galbart. A special thanks to my producers, Cesar Webb, Jim Frank, Lucas Stafiniak, Tom Vanagdam, Bernard Hugni, Curtis Dixon, Benedict Mueller, Vega Guidi, Thomas Trumbull, Catherine and Patrick Tobin, John Carlo Montenegro, Robert Lewis, and Alni Ortiz, and to my executive producers, Matthew Lavender, Sergio Quadrian, and Bogdan Kanivets. Thank you for all.